Let's look at some easy notes that are very crucial in answering questions related to DNA structure in an exam. The DNA is contained in the nucleus, whereby it is arranged in a long molecules called the chromosomes. The DNA was first determined by Watson and Crick in 1953. It is made up of two strands that are wrapped around each other with a double helix, coiled around the axis mainly for protection and stability. The DNA is a polymer as it is made up of monomers, whereby each monomer is called a nucleotide. So it is also called the deoxyribonucleoside monophosphate, covalently linked by 3' to 5' phosphodiester bonds. Nucleotide is made up of 3 parts or components connected to a sugar, whereby this sugar is a pentose sugar, whereby on the other side we have a base and the other side we have a phosphate connected. Every nucleotide has the same phosphate group and a sugar, whereby this sugar, although is a pentose sugar, but then it is called a deoxyribose sugar, mainly due to the lack of oxygen group on one of its carbon. So this is the main reason why DNA is not prone to alkaline hydrolysis like RNA. Phosphate group always bind on the 5' of another sugar, and always note that this phosphate, it is the one that determines the nomenclature. The nucleotides are linked together by the 3' to 5' phosphodiester bond. So what they are trying to say, or what I mean here, is nucleotides bind using phosphodiester bond. So in the DNA structure, the DNA is paired in antiparallel. 5' end of one strand is paired with the 3' end of another strand. The DNA consists of 4 different types of bases, namely the adenine, the thymine, the cytosine, and the guanine. So you need to note that complementary bases always pair with each other. Adenine always pair with thymine, held together by the hydrogen bonds between them, and the cytosine always pair with the guanine, held together by the 3 hydrogen bonds. So the strands of DNA are held together by hydrogen bonds, which are non-covalently, as well as van der Waals attractions or bonds. So these bonds are very difficult to break, depending on the numbers of bonds holding the strands together. For an example, we said that the adenine and thymine are held together by 2 hydrogen bonds, and cytosine and guanine are being held by 3 hydrogen bonds. So meaning that the cytosine and guanine are hard or difficult to break than the adenine and thymine, because they have 2 hydrogen bonds in them. These nitrogenous bases are attached to deoxyribose sugar by the bonds known as the glycosidic. So we have 2 kinds of nitrogenous bases, which include the pyrenines and the pyramides, whereby the purines are adenine and guanine, consisting of 2 rings in their structure, and pyramids are thymine and cytosine, consisting of 1 ring in their structure. So you have to go and look at the 2 types of the structures of purines and the pyramids. That is when you are going to see that there are 2 rings in purines and the 1 ring in pyramids. So another thing to note is a sugar and a phosphate. They alternate to form the DNA backbone, and therefore phosphate acts like a connector between the sugars by forming the phosphodiester bond between the 3rd carbon atom and the 5th carbon atom of the next sugar. This phosphodiester bond is formed when exactly 2 hydroxyl groups in phosphoric acid they react with a hydroxyl group on the other molecule, forming ester bonds, and it also assists with stability and integrity of the DNA. So in this DNA, each strand has a 5' with a terminal phosphate group and a 3' with a terminal hydroxyl group. So when this DNA is being twisted, it forms what is known as the minor and the major grooves. The minor and the major grooves. So these grooves, they provide access for binding of regulatory proteins along the DNA chains. They are also used to assist in the development of different types of drugs, which then binds to interfere with the DNA synthesis and also used in cancer. In cancer, for those who are doing medicine, they will pursue in reading it, or you can follow me for more videos like this. So when we are talking about the supercoiling of DNA, we are talking about the reduction of space and allocation for much more DNA to be packed. So the positive supercoiling, meaning that the DNA is twisted on the same direction, and the negative supercoiling, the DNA is twisted on different direction.